The next slide illustrates the fundamental anatomy of the respiratory or the gas exchange portion of the respiratory system, and this is certainly at the level of the alveolus. The alveolar lining, much of it is formed by the squamous type 1 pneumocytes, and periodically those type 1 pneumocytes are interrupted by the more cuboidal-shaped type 2 pneumocytes. Note that it is the cytoplasm of the type 1 pneumocytes that form an integral component of the blood-gas barrier. And as you note on here, and we'll see this again later, inside the lumen of the alveolus we might find alveolar macrophages, whereas alveolar macrophages may also be found in the thin connective tissue septa that separate individual alveoli. So again, details of the anatomy of the type 1 and the type 2 pneumocytes. Type 1 pneumocytes represent only 40% of the alveolar lining cells, but because of their squamate nature, their thin cytoplasmic processes actually cover up to 95% of the surface of individual alveoli. These cells are postmitotic. Type 2 pneumocytes constitute about 60% of the alveolar lining cells, but they form only about 10% of the surface lining because of their size and their cuboidal nature, and they serve as stem cells not only for themselves, but for also the type 1 pneumocytes as well. Next is a picture of the details of what actually makes up the blood-gas or blood-air barrier. Note that it is formed by the cytoplasmic processes of a type 1 pneumocyte. In addition, that cytoplasmic process is covered by surfactant. Surfactant, as we'll see, prevents alveolar collapse during expiration. The type 1 pneumocytes share a common basal lamina with the endothelial cells that line an adjacent capillary wall. Now, a histologic picture of the wall of an alveolus. And first, certainly we see the three different cell types that are here. Number one, we see type 1 pneumocytes with a flattened elongate nucleus that's characteristic of them. That's at the arrowhead. The rounded profile of a type 2 pneumocyte is seen at the arrow, and in the apical cytoplasm of that rounded type 2 pneumocytes, we can see small vacuoles which basically contain surfactant that's washed out or eliminated during histologic processing. The curved arrow indicates the probable location of an alveolar macrophage that's embedded and has migrated into the alveolar wall. Now the details about surfactant. Surfactant is essential in that it maintains normal respiratory mechanics by preventing alveolar collapse during expiration. Its production in fetal life is essential for survival of an infant that's born prematurely. Surfactant is composed of phospholipids and surfactant proteins whose function it is to spread surfactant all the way across the entire alveolar air-water interface. Surfactant is recycled either back to type 2 pneumocytes, although some of it undergoes phagocytosis by the alveolar macrophages. Surfactant is synthesized, we think, largely after about week 35 of gestation. Corticosteroids can induce the fetal synthesis of surfactant in a premature infant. However, infants of diabetic mothers are going to have a higher incidence of respiratory distress syndrome because of the effect of lack of insulin on corticosteroid and therefore surfactant production. Our last slide summarizes the functions and locations of alveolar macrophages. Like other macrophages, they are derived from blood-borne monocytes that then exit blood vessels and enter lung tissue. The resident alveolar macrophages can lie inside the lumen of an alveolus. They can undergo limited mitoses to form additional macrophages. They patrol the alveolar surfaces, absorbing particles that make it down into the alveolar area. They're also involved in the recycling or take-up or phagocytosis of surfactant. They can pass through pores of cone that connect adjacent alveoli. And logically, they represent the last defense mechanism of the lung because they trap basically inhaled particles at the level of the alveoli. The alveolar macrophages can pass out of the alveoli by passing through terminal bronchioles, bronchi, and the trachea, and or can enter the lymphatics, and lastly, they can become trapped in the alveolar wall with their digested or partially digested material. Alveolar macrophages are also known as dust or heart failure cells.